So yeah, I want to talk about some extraordinary mammals. These are the egg-laying mammals, which I think makes them extraordinary already, but there's so many things about, that are fascinating about them. So this includes the platypus that you see on your left, aquatic animal, and the echidnas, and that's the short-beaked echidna you find all over Australia, the platypus in some regions in Australia, but it also includes the long-beaked echidna, which is only found in Papua New Guinea. They both lay eggs, but when the eggs hatch, they actually feed their young with milk, but they basically just sweat out the milk onto the surface, skin surface, because they don't have nipples. And I could just go on and on. No matter what you look at in these animals, they're just absolutely weird and fascinating. And I think what I really want you to take away from this is when you look at their molecules, if you look at their cells, we just find exactly the same. They're just extraordinary and absolutely fascinating. But in addition, I think to our interest to just learn more about these animals, I think we can actually learn a lot about ourselves, and that really relates in particular to sex chromosomes, where our own sex chromosomes come from, and we had some very powerful insights from studying monotremes. But also I think in general for mammalian evolution, we even use them to find some regions in the human genome that are relevant for diseases. And I think also, and where sort of the passion comes in, these are unique, iconic mammals in Australia, so we always also feel to give something back, to create some tools with the work that we are doing, that we can better understand the ecology, probably even contribute to conservation or understand some of the diseases better that they can be affected by. If you think history is boring, you should really look at monotreme history because it's extraordinary, it's very entertaining and quite dramatic, and books have been written about it. So the first platypus specimen arrived in England in 1798. They were sent by John Hunter, the governor of Sydney. Now, they didn't quite look as the platypus that you saw in my first picture. They probably looked a lot more than the brownish thing that you see on your right in this cartoon. So it was obviously fixed. And the first thing that scientists in Europe were trying to do is to break the beak off, because they thought it was a colonial hoax and it was a beak stitched onto a water rat or something. So when people had realized that this is actually a real animal, it sparked huge controversy over Europe over many decades to how to really explain this animal. And while scientists in Europe were sort of developing their ideas, and one of the ideas really depicted in this cartoon is that the platypus is actually a hybrid between a duck and a water rat. It took another 80 years to actually prove that they would lay eggs, and again, sort of, it was actually then 8 years after scientists sort of got the first specimen, was in 84, that William Caldwell up in Queensland and Wilhelm Haake here in Adelaide that reported independently on the same day to scientific societies that platypus and echidnas lay eggs. Wilhelm Haake in Adelaide, he had an echidna, and he was so excited to find an egg in its pouch. Echidnas have a pouch, that he actually squashed it, and he was devastated. But there wasn't a little echidna in the egg. It was actually off, but I mean, still, he just. So then, in the 60s and 70s, we had the first studies on their DNA and chromosomes, then so I'll talk a lot more about that. But I think again, sort of, and if I get the next. Must be a big picture. In the 80s, then there was some other anatomical aspects in. I don't know if you can get the next one up. That just shows that, again, in different areas, they're just so fascinating. So what they discovered is that the sperm that you see here on an electron micrograph, which actually look like chicken sperm, they form bundles of hundreds of sperm, and they swim together. And I think what advantage is it gives them, we're not quite sure. Maybe they swim faster. The other thing was also in the 80s that scientists were really fascinated by is that the platypus and also the echidna can actually sense fine electric currents. And I think actually in the initial work, batteries were involved, and scientists could show that the platypus would swim towards a battery. So these electrosensors that they have around their beak helps them to find their prey in the murky water. So it could be little worms or shrimps sending out little electric signals by twitching of muscles. So I think, again, it sort of busts the myth that they're primitive animals. They're highly sophisticated and very well adapted to their lifestyle. So I came to Australia in 2001, became involved in platypus sex chromosomes, and we discovered an extremely complex sex chromosome system, and I'll talk about that. Also in 2004, then the Platypus Genome Project started. You probably have heard about the Human Genome Project, so the entire genome of the platypus was sequenced, and I'll talk about that. So I think there's still a lot of work to do. We would also like to get the sequence of the echidna and many other things, but I think we have now a fantastic resource, and what we've seen really is a huge increase in interest and research that is a result of these resources. So I've started talking already a little bit about DNA, genes, genomes, and chromosomes, so I just want to briefly introduce them. So if you look at a cell, and our bodies are made out of billions of cells, we get this nucleus within a cell, and the nucleus is really an amazing part of a cell because it has to really harbor about 1 meter of DNA, and we have 2 copies, so 2 meters of DNA in each cell. Now this is really equivalent to trying to stuff 10,000 miles of spaghetti into a basketball. And the other thing is that it's not just stuffing all this material into the nucleus, it has also to work. So there's probably about 20,000 genes and a lot of other things that are quite important for the genome to work, and they have to all interact in a meaningful way. So it's a huge challenge. But if you want to, and we can get DNA from dead cells and we can study it, but if you want to look at chromosomes and they become really visible, where the genome becomes visible basically in a short window during the life of a cell when it wants to divide, we need to actually living cells. How do we get them from the platypus? So we're getting material from people doing field work. They're catching a platypus to measure it, to weigh it, and they got this massive tow web that they use for swimming, and they can just sort of take a little bit of their tissue that doesn't do them any harm. They send us the tissue in a medium. We can put that into a petri dish, and then cells will actually grow. And I don't know if you see that with the black lump, and you see these little cells growing around them, and that gives us the living cells that allows us to actually look at their chromosomes. So as I mentioned before, when the cell divides, the genome becomes visible as these distinct chromosomes, and of course for many, many decades that has been the almost only way to look at a genome. So here you can see all these little black dots are chromosomes, and here you can see another cell, and you probably think, oh, they look quite similar. These are actually the chromosomes of a human, 46 of them, and these are the chromosomes of the platypus, and there's 52 of them. And you think, oh, they probably look pretty similar. But the main mystery really comes when you're starting to analyze that. So in humans and all other mammals, what we get is that we get a set of chromosomes from your mother and a set from your father. So each chromosome has 2 copies. So you can start searching them. It's a bit like a matching game. And as you can see here in the platypus, you can find pairs for most of them. But as you can see in the bottom row, there's a leftover chromosomes that look all quite different. And people were for many decades puzzled about what they actually are. But the tools weren't really available to identify them. So when I came to Australia, we were applying a technique that was developed some 20, 25 years ago, which basically allows you to label a piece of DNA with a fluorescent color, and then we can use this amazing ability of DNA to find its complementary sequence. So we can use this DNA, we can basically paint it in red or green, we put it back onto the chromosomes, and it will find its identical, basically, partner, and therefore, as this X-shaped chromosome, you then get a nice signal that you can see under the microscope where this piece of DNA is located. You can do that with the whole chromosome, and then basically talk about chromosome painting because the whole chromosome is stained. And this is an example of the experiment that we did. So you can see on the right in blue these chromosomes, and then you can see these green areas where the DNA that we have painted in green was basically identifying its identical sequence on the chromosomes. So we did that for all the chromosomes, and what we identified was more than surprising, because humans have 1 sex chromosome pair. In platypus we found 1, but that wasn't enough. We found another one, and I'll just keep clicking because there's a few of them anyways. And hopefully we'll get to that. Okay, so I'll just keep clicking. So it's 2 of them, 3, 4, and another one. So 5, I'll just go back. So there's 5 sex chromosomes in the platypus, and that's world record. There's no other mammal that has 5 sex chromosome pairs. And when we published that, of course, that sparked a lot of interest, and people kept asking, why do they have 10 sex chromosomes? Is that cool that you have 10 sex chromosomes? Is that something I want too? But I think what I want to share with you, it comes with a few difficulties. And that's really what we, I think, trying to get our head around now, is really to understand how they deal with it. Because I can't answer the question why they have have 10 sex chromosomes, but I can tell you that they're very good at dealing with them, and they have no problems in sorting them out. And my next slide is showing exactly that. Just to give you an appreciation that this is actually quite a difficult thing to be dealing with. In humans, in males, if you want to make sperm, we have an X and a Y chromosome. The Y chromosome is male specific, and the X chromosome goes to one sperm, and the Y chromosome goes to another sperm, and that should actually, there it's in pink, here it's in blue. Now, if you're a platypus, this becomes a lot more complicated because you have 10 X and 10 Y chromosomes. What do? You don't want an X and a Y chromosome in one sperm because then you can't get males and females. So what they actually do is they put them together in a little chain where they are arranged in an X and a Y chromosome, and then they manage somehow, and I'll just keep clicking, managed to get all the 5 X chromosomes in one sperm and all the 5 Y chromosomes into another sperm. And we know it works really well. We've looked at hundreds of they never get that wrong, but we still have not a very good understanding of how they actually do that. So that's really the work that we're doing at the moment. So another thing, and that's really sort of bring this part of my talk where we can learn from the platypus. So this is an extraordinary system, and I think we're just absolutely fascinated with it and want to know how it works. But what does that mean for humans? And we really came to that when we were starting looking at the genes on these sex chromosomes. And that really didn't bust a few myths, because when we looked at the genes on platypus sex chromosomes, we found that they're not on the sex chromosomes in humans, but they are the genes that are on sex chromosomes in chicken. And before we were thinking that all mammals have the same sex chromosomes, so that's wrong. We also thought that birds and mammals have basically evolved their sex chromosomes independently, but now birds and platypus seem to have the same sex chromosomes, so that wasn't true either. So what we're currently thinking is that the ancestors of mammals and birds had the same sex chromosomes. They were maintained in birds and monotremes, in the platypus and the echidna, and after they diverged from the mammalian lineage, marsupials and placental mammals, that's the other major groups of mammals, evolved another sex chromosome system. So for us, this means that our sex chromosomes are a lot younger than people have previously thought when they thought that all mammals had the same sex chromosomes. So now I want to really go into the Platypus Genome Project. Why would you want to sequence the platypus genome? And I've already mentioned a bit of the fascinating biology, electroreception, the 10 sex chromosomes, and so forth. And so I'll just skip over that. And just want to remind you also of the importance in evolutionary sense. If you look at the mammals, we got the placental mammals, which are mouse, dogs, humans, cows. We got the marsupials, kangaroos, wallabies, and then we got these monotremes. And it was really a thought that we need to get genome sequence from all these groups, as well as many, many other groups of plants and animals. And I think it also illustrates that they, evolutionary on this tree, basically sit really nicely between the birds and the other mammals. So again, for comparisons, very powerful. So it was decided that the platypus genome was worthy of sequencing. These are huge projects, cost several $1 million, and there was over 100 researchers worldwide involved in this. So a lot of international interest also in the species. And again, comparing genes in different species gives us really powerful insights on how these genes have evolved at different functions. And that's important for understanding the origin of our own genome, but actually also how things can change, for example, in human disease. So in 2008, the platypus genome was basically finished and published, and sort of made the front page here in the Advertiser, which was very good. And just to give you a bit of a feel what that actually means, a genome project. And I sort of, to illustrate that, I looked up how many genes did we know before the genome project started. That was a total of 23 genes. And that was between 1974 and 2004. So in 30 years, 23 genes had been discovered. So that's a rate of less than 1 gene per year. And in fact, it could take a student up to a year to actually find a gene. The Genome Project described over 18,500 genes, which is pretty close to the estimated gene number in humans, which is about 20,000. So you can just see that jump from almost less than 1 gene per year to 4,500 genes per year. And now we got them all in front of us. We can look at the computer. Everybody in the world can look at the computer and find the sequences. So I think after that was finished, 2008, we had a great conference here in Adelaide, over 60 people, also from all over the world, but a lot of Australian researchers to really get our heads together and think, what are the next steps, what are the next challenges, what can we do with all these resources and new knowledge? And obviously we want to understand their extraordinary biology, But as I said before, I think we also want to probably develop tools that can be used in ecology, probably even in conservation, also to understand some of the diseases that can affect monotremes, but also these insights that we can gain about mammalian evolution. And that's certainly what people interest worldwide, and that's why the platypus genome was sequenced in the first place. So I just want to give a snapshot, and I'm not sure how I'm going for time, to some of the projects, again, where we're trying to take some of the tools that we're developing into the field. So we're having one project, it's a long-term project we're having up at Monado Zoo, where there's probably about 30 to 40 echidnas, and when somebody sees an echidna, they grab it, we put a radio transmitter on it, so working closely together with very enthusiastic volunteers, and here's a picture of a volunteer with the echidna. I don't know if you can see in front with the radio transmitter. We also get, again, our little tissue piece, so we can grow the cells in the lab, and so that we can do genetic studies. And what we're hoping with this is that we can look at things, how many males and females are there around? Is there some males that are reproductively more successful? But there's also a number of ecological questions that are really fascinating in that particular environment. But we've also, and we're looking for male-specific genes in the platypus, and we've basically used that to develop an assay where we can genetically determine the sex of animals. And especially in the echidna, this is actually not a trivial thing. And so we got in contact with people at Perth Zoo, where they've been quite successful in getting echidna offspring. You can see these beautiful puggles here on the left side from Arthur Ferguson at Perth Zoo. And they pulled out a few hairs, and the hair roots have so many cells that have DNA, we can isolate that, and then we could do tests to determine whether that contains male or female specific DNA. So here you can see that we've been able to detect with the genetic method male specific DNA, so we know this is a boy. But when we looked at all the puggles from Perth Zoo, it actually turns out that they're all girls. Why that is, if that's just sort of random or if there's something else to it, we don't know. But that was the result, and I called Arthur up and I said, look, I think our test is probably not working, they're all females. But he said, well, when I sort of looked at them, he thought that might be right. Okay, so I just want to go to my last slide, and there's one I think where we're interested in to understand why platypus in Tasmania are particularly susceptible to a really nasty disease, which is called mucormycosis. It's called, or fungal disease, it's caused by a fungus, and they get these really nasty ulcers that you can see here, and eventually they die. And it's a major mystery why it's mainly, or almost exclusively, Tasmanian platypuses that are affected. Is it because the fungus is more virulent in Tasmania, or has it something to do with the population, even with the genetics or the environment? So we're trying to understand what are the genes that are involved in the development of this disease, and in the future we also want to look at the fungus at the same time to really trying to get a better understanding of that disease. So what we're really passionate about, I think, and we're excited about, is that there's a new era in monotreme research. We have now these amazing resources, and we have also this worldwide interest in the monotremes, and we want to use that, obviously, to understand their biology better, but also, I think, use their ability to give us fascinating and new insights into mammalian evolution and disease. But again, I think we're passionate about and aware of the fact that we are in Australia, where these species are, and we have a special responsibility, I think, to ensure the survival of this unique species in the long term. So I think with the little bit that we can do in our research area, we always sort of keep an open mind if you can actually develop some tools and research that will actually assist in this research and also hopefully help preserve them. We're very grateful for funding from the Australian government, which I've received now for almost 10 years, and I'm extremely grateful of having students that are just as passionate about this research as I am. Thank you.